research on the condensing and fluidizing effects of gangliosides and phospholipid films. It sounds a little challenging, but really when you work it down, it's pretty simple. So all cells have a membrane on the outside of them, and this membrane has a lot of cool dynamics and fluids to it. So recent research has shown these lipid rafts to exist in membranes. These are like little areas that are more condensed. And we try to study what these are because we found out that they're very, very important to what the cell does in terms of where proteins are in the cell outer layer. So what our group does is we basically put a membrane on a little fluid of water, and we try to study the properties by kind of condensing it and expanding it. So previous research showed that with a phospholipid and a ganglioside, we see that the two of them become more condensed. And so what my research was to see how changing the phospholipid actually can affect what happens with this condensing that we see. And so over the course, we changed the phospholipid several times to see how the size, the shape, and the charge all affect the properties. And what we end up seeing is that regardless of whether or not we change the size or the shape, or even the charge, we can still see the same condensing effect that we can see in cells. So hopefully if we take this, we can learn more about how cells work and kind of map out what we can do in the future.